Bananas, Musa subspecies family Musaceae, are giant perennial herbs growing in tropical and subtropical regions of Asia, Africa, South America, and Australia. Being an essential nutrition source for millions of people in developing countries and one of the top export commodities, bananas are one of the world's most important fruit crops. The global banana industry started in the late 1800s as a result of high-tech advances such as refrigeration on ships. The fruit itself is high in essential B vitamins, and their leaves are proving useful as hygienic plates in many Asian countries.
Polyploid bananas, specifically triploids, followed the origin of diploids. In nature, positive natural selection favoured the heterozygotes that were almost or completely seed sterile. Cultivation and a non-preference for selection of seed-fertile clones over time have led to the collection of structural changes in chromosomes. The selection was generally due to the appeal of bananas, such as texture and flavour, as well as their suitability to production, including bunch numbers. The variety of banana best known to us today is the Cavendish, named after the Englishman William Spencer Cavendish, the 6th Duke of Devonshire. In the 1900s, Cavendish became one of the world's most popular banana varieties and remains so today. It is one of the most resilient banana varieties, as the plant is resistant to some soil fungi, which can harm or destroy other banana varieties. All Cavendish bananas are genetically identical and clones of one another. They are sterile and do not have seeds.
Most wild bananas are diploid, which means they have 2 sets of chromosomes. However, commercially grown bananas are all polyploid, more specifically triploid, meaning that they have 3 sets of homologous chromosomes. Normal meiosis on a diploid individual involves DNA replication followed by 2 rounds of cell division to produce 4 daughter cells, each with half the number of chromosomes as the original parent cell. A gamete would then receive 1 haploid chromosome from each parent. But how do polyploid plants, or plants with more than 1 set of chromosomes, form? Polyploids arise when a rare mitotic or meiotic catastrophe, such as non-disjunction in meiosis 1 or 2, causes the formation of gametes that have a complete set of duplicate chromosomes. Diploid gametes are frequently formed in this way. When a diploid gamete fuses with a haploid gamete, a triploid zygote forms.
Bananas are monoecious, meaning they contain both the male and female sex organs on the same plant. Female plants are within the basal or proximal bracts, and male flowers are in with the apical or distal bracts. The male part of the plant produces pollen, which fertilizes the female flower of another plant. The ovules must be fertilized within 24 hours of the flower opening, after which they begin to disintegrate. Banana plants are also dichogamous, meaning there is also a temporal as well as a structural separation of the sex organs, i.e. the female organs mature before the male organs, and as a result, self-fertilization is prevented. Parthenocarpy refers to the phenomenon in which a plant is able to produce fruit without fertilization. Bananas have vegetative parthenocarpy, which is the formation of fruit without the requirement of any external stimulus, i.e. pollination. As a result of parthenocarpy, the fruit often has no seed, which is desirable for consumers. However, the lack of seeds can often be misblamed on parthenocarpy, when it can also be a result of polyploidy, as triploids especially produce few seeds due to reduced fertility. This is the result from faulty chromosome pairing during meiosis, so triploids are highly sterile.
Due to their susceptibility to pathogens that comes from lack of genetic variation, introducing new genetics to the commercial banana crops is incredibly important. Unfortunately, the polyploidy of the commercial banana crops results in sterility, making it difficult to improve. Most breeding programs aim to improve fruit yield and resistance to diseases such as black sigatoka, whilst maintaining the quality of the fruit. The current main method of crossbreeding to improve the banana crops can take up to 15 years due to the time taken to grow trees to fruit-bearing age. It starts with crossing a sterile triploid variety with a fertile diploid. The tetraploid progeny taken from this are then crossed with superior, more commercially viable diploid hybrids, and the triploid progeny from this crop can then be selected for commercial production following strict evaluation. As this is a lengthy process, researchers are exploring ways to refine it through things such as genomic selection. This is the use of genetic markers to improve the occurrence of desirable quantitative traits in the banana crop, such as resistance to the black sigatoka, with a greater accuracy, as well as enabling the visualization of genetic diversity amongst the banana crops.
Before the Cavendish, Gros Michel bananas were the main commercially grown banana. In the 1950s, it was completely wiped out by Panama disease, a wilt caused by a fungus. Cavendish proved resilient to this and hence is the banana we see today. There are many pathogens that threaten the banana plant, and due to their genetic similarity, there is the risk that all plants in a plantain will be wiped out. If there's a lesson to be learned about bananas, it's that reliance on a large and genetically uniform monoculture is a risky strategy that is prone to failure due to the risk of wipeout from disease. To reduce the vulnerability to diseases, we need more genetic diversity in our cultivated bananas.